Hey guys, here is a whole of B2 for AQA. You need to know the structure of animal cells and what all the different bits do. So this big blob in the middle here, this is the nucleus. That is where the DNA or the genetic information is going to be held, and this is in charge of that activities. Then we have these smaller blobs. These are the ribosomes. These make proteins. These squiggly bits are the mitochondria. And they are in charge of releasing energy, and this is where respiration happens. Tucking this all in, going all the way around the outside, we have the cell membrane, and this just holds things in. And it also controls in and out. So control stuff that goes in and then control stuff that goes out. And then all around the kind of like gooey stuff that is left, this is the cytoplasm. And this is just where stuff happens. It contains a lot of enzymes and it's kind of just there. Plant cells have quite a few things that animal cells don't have, but if we start with some similar things, so we have the nucleus, and again that is where all the DNA is held. Here we have the mitochondria. This is again where respiration takes place, where energy is released from, the ribosomes. Again, these are in charge of making proteins, and then this inner one here is the cell membrane, again in charge of what goes in, what goes out. Now the other things that they have are on the outside, so the outer one, this is the cell wall, and this is very hard in comparison, very rigid, and this is what gives it its shape. So it keeps the shape really well. We have chloroplasts. These are for photosynthesis. We have this big gap in the middle. This is the vacuole. And not a lot happens here. It just contains a very weak solution of sugar and salts. We have our very small, very simple yeast cell, but astonishingly important. It has some similar features to plant cells, like the cell wall, and then on the inside the cell membrane. It has the cytoplasm and a nucleus. And then our bacterial cell, and again the inner one is the cell membrane. The outer one is the cell wall. In the middle we have our cytoplasm. And no nucleus, but we have our DNA kind of floating around in the middle there. They love asking about structure and function of the different parts of cells, so make sure you learn this bit really well. There are several different specialised cells that you need to know about. So we have sperm and egg cells. Now these are obviously for reproduction, and if you think about what a sperm and egg cell actually has to do, it has to do very different things. So the egg cell has to kind of, well, sit around waiting to be found. But this little sperm cell, and to be honest, the sperms are tiny, has to do an awful lot of swimming. So it has just behind its head this kind of like little band which is chock full of mitochondria. Because if you think about it, anything that has to swim for a very, very long way or do a lot of stuff is going to need a lot of energy. Energy takes place from respiration, which happens in mitochondria. So they're going to need a lot of mitochondria. So that is why sperm cells have a lot more mitochondria than other cells. And when you think about the shape of it, it's very, very streamlined so that it can cut through everything it needs to when it's swimming. Whereas the egg cell is very, very large, and in the middle it's going to have lots of nice gooey stuff. So it has kind of lots of food reserves just waiting to feed the developing embryo. Red blood cells are again very, very a specialised cell. They have no nucleus. And the advantage of having no nucleus means they have more space to carry oxygen. They have this kind of curved shape here, which means that it does have a lot of surface area, and it's not just on one side, it's on both sides. So we can call this a biconcave shape. And the other things that they have in there, which they need to carry the oxygen around, is the hemoglobin, which gives blood its distinctive red colour. Interestingly, the red colour comes from the iron in hemoglobin, and horseshell crab's blood, their hemoglobin takes up copper, so their blood is blue. Horseshell crabs are incredibly interesting things. Yes, and red blood cells are very, very small, they're tiny. Now, if we move on to thinking about specialist cells in leaves, the palisade cells, which are going to be in the leaves, so they're all at the top of the plant, which is where the sun's going to be, and they have loads and loads of chloroplast in there, because that's the bit where photosynthesis takes place. Now they have a lot of surface area. Anything where there's going to be absorption or diffusion or something going from one side to another side is going to need a large surface area. So the definition of diffusion, because this is important, they could ask this as a keyword, is spreading out of particles from places where there is a high concentration to places where there is a low concentration. So what this means in air, if we start off with a high concentration of purple particles down here, they are going to want to spread out to this area of low purple particle concentration over here, so that they are all nice and neatly spread out. If we have a cell membrane involved, again, the purple particles are going to be the ones that want to move, and what they want to make sure is that there are an even number of particles on each side of the membrane. So we have an equal concentration, so things have moved from a high concentration to a low concentration. So a tissue is a group of similar cells. And when they all get together, lots of different types of them, they become an organ. And when we have lots of different organs all working together, it is an organ system. One example of an organ system is the digestive system, where we have lots of different tissues working together, lots of different organs all working together to perform one overall function. So we have the large intestine, which reabsorbs water. We have the small intestine, where food and sugar and various different things like that are reabsorbed. We have the stomach, where the food is digested. We have the liver, which adds in things like bile, which helps to neutralise the stomach acid, and then we have the mouth, which breaks down the food. So lots of different organs, the liver, the stomach, the large intestine, all working together in an organ system. Plants have organs too. The organs that they have are leaves, they have stems, and then all the way down the bottom they have roots. So they're the organs that a plant can have. The tissues that a plant can have are the mesophyll, and this is where the photosynthesis takes place. We have the xylem and the phloem. This is for transport. So sending things like sugar, water, ions that it needs all around the place, and the epidermal tissue. This is like the skin of the plant. Photosynthesis is the method that plants use to make food. They take in carbon dioxide, and water, and convert it into glucose and oxygen. Now for this to happen they need light. Now the reason we write it above the arrow like that is because we need it to happen, but it's not actually a reactant, so it doesn't get used up in the equation. Now there are several different things that a plant needs to photosynthesis to take place, and we can call these limiting factors. So the limiting factors for photosynthesis are going to be light. So if we don't have light, we're not going to get a lot of photosynthesis, so at night. The reactants. For example, if we don't have any carbon dioxide or any water, then we are not going to get photosynthesis taking place. And the other thing is also going to be temperature. When we're looking at the rate of photosynthesis and light intensity, from a point when there's no light, there's very little photosynthesis or no photosynthesis, it is going to increase steadily up to a point where it is no longer the limiting factor, where something like carbon dioxide, water or temperature is then going to be the limiting factor. When we're looking at how carbon dioxide affects the rate of photosynthesis, it is going to go up steadily and then level off when something else, like light, becomes the limiting factor. The graph of how temperature affects photosynthesis is quite different from how carbon dioxide or light intensity affects photosynthesis. When we have quite a low temperature, we have a slow but steadily increasing rate until we get to the optimum of the peak temperature. After this, it is going to decrease really quite rapidly until we get to a point which is about 45 degrees where the enzymes are going to be destroyed. And once this happened, once the enzymes have been denatured, once they've been ruined, they cannot function anymore. So there is an optimum temperature for photosynthesis. Below that, things will just work slowly. Above that, things just won't work at all. The glucose that plants make in photosynthesis is used for many different things. Now, believe it or not, as well as photosynthesis, plants actually undergo respiration. This is the same equation, just in the reverse direction. They also use the glucose for building structures, so like cell walls. And they use it for making proteins. This glucose can be stored in two different places. It can be stored as oil, so that we get out of the crushing of the seeds of the plants, and this is where it's going to be stored as a lipid, or it can be stored as a carbohydrate, so it can be stored as starch. And these are going to be things like your big fat potato tubers, the roots, the stems, the leaves, the seeds, loads and loads of different places that starch can actually be stored in plants. When we're talking about where a living organism lives, there are a couple of words that we use. We use habitat. That is just the place that it lives in. And we use distribution. So that is where it's found within that particular location. And there are going to be a number of things that are going to affect distribution, whether it's distribution of the grass, whether it's distribution of the trees, or whether it's distribution of the animals in there. And these things are going to be temperature, the amount of water, oxygen, carbon dioxide, nutrients, and light. Because whether you're an ape, whether you're grass, you're going to need to be near somewhere that has water. You're going to need to be somewhere that has the right levels of oxygen, right levels of carbon dioxide, where you can get food from, where you can get light, and where it's not too hot or not too cold for you. Now, when we are looking at an area, when we want to study this distribution, we can work out what lives there in a couple of different ways. And the first way we can do it is with quadrats. So what you need to do is to get your quadrat and place it randomly. So basically chuck it in a field somewhere. You are then going to need to count everything in the square. And because because we're scientists, we don't just do one things, we do this 3 times. We are going to need to repeat our counting. Once you've repeated our counting, we need to work out a mean, and then we need to move the quadrat to a different location within the area. Again, we're going to need to count stuff, repeat the count, work out an average, and then once we've done that, we can compare the distributions. Proteins are long chains of amino acids. And amino acids are like the building blocks, the bricks of absolutely everything. And proteins go on and do loads and loads of really, really useful functions. So they build up the bulk of everything. So they are your muscles, all the bits in tissue that need building up, that's what they do. They are your hormones. They are antibodies. And they are your enzymes. And the enzymes are incredibly busy going around your body doing loads and loads of things. So what is an enzyme? Well, an enzyme is just a biological catalyst. Which means it speeds up a reaction. But stays the same at the beginning as at the end, so it doesn't change or get used up. Enzymes work by what's called a lock and key mechanism. Only one thing is going to fit into the space. So here we have 3 substrates, and you can see substrate number 1 is going to come along, it's going to try and fit in there, but it doesn't fit in there, so nothing is going to happen. So this one is just going to go off now. So substrate number 2 is going to fit in there, can't fit that way, turning around, can't fit in that way either, so that one's not going to work. Substrate number 3 comes along and shock horror, it fits in absolutely perfectly. So now we're going to have a reaction taking place. So now the substrate's fitted in, the enzyme can work its enzyme magic and actually do something, either break something down or put things together. But if the substrate doesn't fit in there, nothing is going to happen. It's only if the substrate fits in perfectly will something happen, which is why it's called a lock and key mechanism. Enzymes have an optimum temperature that they like to work out. Now in our body, that is 37 degrees, but you can get extremophiles, which like to live at really, really hot or really, really cold conditions. Now below that optimum temperature, they're going to work, but they're not going to work very well. They're going to be slow, they're going to be lethargic. And they get to that optimum temperature, they're going to work really, really well. And then very quickly, they're going to go down until this point here at which the enzyme is denatured. We don't say that an enzyme has been killed, we say it has been denatured. Say it is killed suggests that it was alive, but all that's happened is changing shape. The bonds that hold it all together have been broken, which means it's not going to work, so it has been denatured. The same idea applies when we're talking about pH, but an enzyme can be denatured by too low or too high pH. So we actually have this very narrow looking curve where either side of it the enzymes don't work, but then in the middle up here we have our optimum. The digestive system is packed full of useful things that do stuff. So the first one that we come across is amylase. and amylase will turn starch into sugar. And this is found in a number of places. It can be found in the saliva, which is why if you put a bit of bread into your mouth and just leave it there, it will eventually, after a while, quite a long while, start to taste sweet. That's because the amylase in your mouth is just breaking it down. It comes from the pancreas and in the small intestine. The next one that we come across are proteases. And they will take a protein and break it down into amino acids. Again, these are found in lots of different places. So the stomach, again the pancreas, and the small intestine. Next one that we have are lipases, and they will break down lipids, which are fats, and they will break them down into fatty acids and glycerol. These can be found in pancreas and the small intestine. And then lastly on this slide, I'm just going to mention bile, because it's not an enzyme, but it is involved in this. And what it does is it's just two things. It neutralises stomach pH, so the hydrochloric acid in the stomach is way too acidic for enzymes in the small intestine to work properly. So you add the bile in once the hydrochloric acid and the digestive goo has come out of the stomach, so that the small intestine is a bit more alkaline so it can work properly. The other thing that it does is that it emulsifies fats. Which means breaks them down into smaller lumps so it has a larger surface area. And the bile is produced in the liver and stored in the gallbladder. As well as being in our body, enzymes are all over our house. They're used for loads and loads of different things. So the most common one that you will know of are washing powders, which is why there's very little point in washing your clothes on a really hot temperature, because most enzymes, as we've seen, are only effective at about 30 or 40 degrees. And they can also be used in industry. Now, they're used for something that sounds really gross, pre-digesting things. So they can be used in baby food, or they can be used in soft scented chocolates. So they're soft scented chocolates isn't actually soft as it goes in there. It's hard, and then the enzymes that are in there slowly break it down over time so it becomes soft once it's encased in the chocolate. Aerobic respiration takes place when there's lots of oxygen around. So we have glucose plus oxygen will turn into carbon dioxide plus water, and most importantly, energy. Now I've put energy in here, even though I'm gonna put it in a little circle, because it's not really a reactant, it's just something that comes out. The reason we do aerobic respiration is to get energy out of things, but it's not really a reactant, so the chemistry teacher in me doesn't really like putting it over there. Now, this aerobic respiration, it's going to take place in your mitochondria. And like I said, this is only going to take place where there is lots and lots of oxygen around. If we don't have a lot of oxygen around, we have anaerobic respiration. And the glucose will then turn into lactic acid and the energy again. Now the lactic acid, you're going to get this when you're doing things like sprinting, running really fast, doing lots and lots of exercise. That's the stuff that hurts. It really, really hurts when you do loads and loads of exercise. That is because once you've stopped, you've built up this oxygen debt. And you have to breathe really heavily because you have to repay this oxygen debt. So blood's going to be flowing around. It's going to want to be removing the lactic acid and turning it into carbon dioxide. We are actually very, very complicated beings. So here we have a cell. Inside our cell we have our nucleus. Inside our nucleus we have chromosomes. I'll just put that arrow there. Inside the chromosomes we have all of this coiled up DNA. And the DNA is made of 2 strands. So this is our double helix, our DNA. And DNA is made up of, as we can see here, bases which are A, T, C and G. And the structure of this, the sentence that it spells, we can then go from DNA. DNA has sections in it which are a gene. You can think of that as like a word or a sentence in a book, and then the sections of DNA will code for a protein. And the proteins are the building blocks of everything. Everyone's DNA is unique, and we can use that for a couple of things, and this is what we call DNA fingerprinting. We can use it for paternity tests, or we can use it for forensics. So if there's blood or something left at a crime scene, we can work out whose it comes from. Mitosis is a process of a cell reproducing itself to form 2 identical cells. So this is the type of cell division that will take place in your skin, that will take place in your hair, when your body is repairing itself, or in asexual reproduction. And what we end up with, what we start with rather, is just one cell which has the DNA and the nucleus in it. It gets a signal that it needs to divide, and all of the chromosomes line up down the middle. Once they're all lined up down the middle, once they're ready to divide, they start cell division. Once they know that each half of the chromosomes is around each end of the new cell, then we can have new membranes forming. and then the cells can start to separate. So we end up with two identical cells that are made from one cell. Now meiosis is something different. This happens in sexual reproduction, so it's going to happen in our gametes. This is going to be our sperm or our egg cells. And what we end up with is 4 different haploid cells. Haploid means it has half the amount of DNA. So it is going to have 23 chromosomes, not the 46 chromosomes or the 23 pairs that you would normally see in a skin cell, for example. Now meiosis, you're only going to get 1 set of chromosomes, you're going to get 4 new cells, and remember this happens in your reproductive organs only. I have a really stupid, silly way of remembering the difference between meiosis and mitosis, and I've made a separate video explaining that for you. Stem cells are fantastic little things, and the really interesting, amazing thing about them is they have the potential to differentiate into anything. This means they can be really, really useful in curing diseases, because we can take cells that can be specific to the person and turn them into any type of cells that we want. So it could be nerve cells to replace people who are paralysed by spinal damage. We can replace cells in tissue, for example, that people that have heart disease, or it can be used in cancer treatment. But there are some ethical issues surrounding this. So this might be one of the questions they might ask you in the exams. There are strict guidelines surrounding the use of stem cells, partly because some of them come from embryos, and partly because some people don't think this sort of research, this sort of treatment should be taking place because of religious reasons. The chromosomes that determine whether we are male or female are our sex chromosomes. So a woman will have XX chromosomes, and a man will have XY chromosomes. Now all eggs are going to be X chromosomes, and then the sperm is going to be the bit that decides whether you're male or female, because they're going to be 50% X and 50% Y. Now there are a couple of different ways that you can draw a genetic cross. I prefer doing what is called the Punnett square. So here we have an RR and an RR person. The first thing we need to do is to work out the gametes. So we're going to have an R and an R and an R and an R. I do understand me saying R and R and R and R very lots isn't necessarily the clearest thing if we're not actually watching the video for this. Then you need to draw out a grid like this, and then put your gametes in the top. So R, 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 and then fill in things. So this column here going across, we're going to have all R's. So R, R, and then the lower one here is all going to be lowercase r's. So R, R, and then going down here we need capital R's. So R, R. And then going down here, we need lowercase r's. So R, R. Now what we have is R, 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 R in the ratio of 1 to 2 to 1. Cystic fibrosis is a disease where the cell membranes are disrupted, which leads to a large number of different phenotypes. So lots and lots of things that are a problem for people with cystic fibrosis. The most common one that people know about is a problem that people have with breathing. Now, cystic fibrosis is a recessive disease. which means two people can have the gene for cystic fibrosis and have absolutely no symptoms at all. And the way, if we just want to draw the Punnett square, that this works is we have the gene for cystic fibrosis, which is a capital F. And then this one here is going to be the disease gene. So our gametes are capital F, lowercase f, capital F, lowercase f, and these are our parents. If we just draw out our Punnett square, capital F, lowercase f, capital F, lowercase f. Now, if you, like me, your Fs look very, very similar, you won't want to consider in the exam just drawing making it very, very clear to the examiner that you can do, so make it abundantly clear that this is a capital F and that this is a lowercase f or something else, because if the examiner's not sure, they're not going to be able to give you the marks. So if we just fill this out, and then this person here who has 2 recessive genes is going to be a sufferer. This person here is going to be normal, and these 2 people over here are carriers. So we can say, if you want to do it to the normal to sufferers, we are going to have a 3:1 ratio. People with polydactyly are going to have extra fingers or toes. And this is a dominant gene. So the capital D is going to be the person that is going to have polydactyly. And then lowercase d is going to be for the normal gene. So if we have one parent that has polydactyly, capital D, lowercase, 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 and they make a baby with somebody who doesn't have polydactyly. Here's our Punnett square, lowercase, lowercase, capital, capital, D, 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 D. We are going to get 2 people that have polydactyly and 2 people that do not have polydactyly. And this is going to be in a 1:1 ratio. Technology has advanced so much that we can actually do embryo screening, which is screening of babies before they're born. So there are a couple of different things we can do. We can do pre-implantation genetic diagnostics, or we can actually sample the cells of the placenta and look at those, which is chorionic villus sampling. So pre-implantation genetic diagnosis is going to be things for like IVF, where you're going to be implanting the embryo, and what you can do is actually just take one of the cells out. This won't affect it at all, and analyse that to see whether it's got the healthy or disease genes, and the ones that are healthy are the ones that can be re-implanted. Chorionic villus sampling is to test for other things where you haven't had IVF, but you want to know if your baby may be carrying some kind of genetic disease, and then with that information the parents can make a decision as to whether they want to continue the pregnancy or not. Now, there are advantages for and against this. The advantages for it is that it stops suffering from babies that are born with genetic diseases. So if you're pregnant and you find out your baby has a disease, which means your baby is literally going to live days after it is born, and it's going to spend that time in large amounts of pain, you might decide that you actually don't want to continue with that pregnancy, and that you as the parents and the baby don't want to go through that suffering. The arguments against this is that leading on to the Gattaca argument, which is one of my favourite films, that's where you're going to get designer babies, where people might decide that they only want to have tall blonde babies, and that any brunettes won't be born, or that they only want to have really athletic babies, or that they only want to have really intelligent babies. So that's where the problems with designer babies are coming from. And with chorionic villus sampling, the problems with that one in particular is that they can lead to miscarriage, which would be awful if that happens and you actually have a healthy baby in there. Fossils are absolutely fascinating and beautiful things, and they tell us so much about how life on Earth evolved. And what they are is the bones, the leaves, the hard solid bits of things that have died and millions of years ago and been compressed in places where no decay happens. So we're going to get lots and lots of soil going down on them, so there's no oxygen in there. And there might be some high temperatures in there, and that's when we're going to get fossils. They're the preserved dead animals and plants. Here is the poor old dodo, which by all accounts was an absolutely lovely bird, but they don't exist anymore because they were hunted to extinction, and humans also introduced loads and loads of other animals which ate all of their eggs. So this poor dodo is now extinct, and there are a couple of reasons that animals become extinct: because the environment changes. You could have a new disease or predator. You could there could be competition for food. Or it could be something happens, so a catastrophe. Now, just as species can become extinct, they can also create new species, which is called speciation, and this can happen due to isolation. It can also happen due to natural selection. Well done for making it all the way to the end of the video, guys. Excellent work. If you've been putting this much work into all of your revision, then I wish you all the best with your exams. Good luck, and don't forget, if there's anything else you need from me, just ask.